This event was made possible thanks to the cooperation and availability always demonstrated by the head of the center, Professor Olga Pom, and also the fine work of Ricardo Santos, the man behind the organization of the conference and the logistics. Thank you, Ricardo. To speak about our guest is not only talking about a world expert on role of virus evolution on life, including its origin, but also about viral gene therapy, cancer virology, biodefense, and proteomics. Luis Villarreal is professor of molecular biology and biochemistry at the University of California at Irvine, and was founder and director of the Center for Virus Research of the same university. From his long and fruitful scientific activity, I would like to emphasize his book, Origin of Group Identity, referring a sentence from it. With the sequencing of entire genomes, and more recently with the shotgun sequencing of habitats, we have come to realize virus are the black hole of biology. A giant force that has until recently been largely unseen and historically ignored by evolutionary biology. Virus not only can cause acute disease, but also persist as stable unseen agents in their host. In this, they attain stability in evolution. It is from such a persisting relationship that virus can inform us regarding the strategies and mechanisms of group membership. I think it is really important, this idea, and I believe that Professor Luis Villarreal will develop it in this communication. Before I give the word to Professor Villarreal, I also would like to remember the words of a philosopher of biology, Michel Spedel, in her article, The Parasitic Host, Symbiosis contra Neo-Darwinism, referring to the traditional criticism associated to this paradigm in evolution. She said, "Nature is competitive and cooperation is a strange case that needs to be explained." In our point of view, and reinforcing Spedel's statement, we do not need to explain and understand cooperation as an alien idea. We should consider it as a synergistic force that is recurrent and central in the evolutive process. In this context, we need to investigate it and consequently incorporate the concept as one of the main scientific principles explaining the origin, organization, and evolution of life. I believe the conference of Professor Villarreal is the best way to understand this issue, beginning by the importance and role of virus in evolution. So, I give the word to Professor Luis Villarreal, who will talk about can virus make us human, addiction and social cooperation as viral. Thank you.
Accepted to come to as a specialist in the area to collaborate with us in the debate, which will be taken after the talk, and also to thank all the young people who is in the room. It's very important to see that the young people is here. It means that the future of science is concerned, because the future of science is always concerned with young people. So thank you very much.
Thank you for inviting me. I'd like to give special thanks to Francisco and Olga and Ricardo. You will excuse my Spanish enunciation. I have sort of an addiction module in my brain that prevents me from learning the Portuguese version of these names. But it's been a beautiful day. This is a beautiful center, and I just hope I can maintain the sort of the sense of the beauty. What I'd like to talk about today is a thesis that emerged over many years having to do with the role viruses play in the evolution of life, a role that's historically been sort of not acknowledged. The specific title of the talk I'm presenting today was actually a title of a talk that I gave some years ago to the American Philosophical Society. And at that time, I'm sure many in the audience thought that this was a preposterous idea, that viruses would have anything to do with the evolution of the complicated phenotypes and characteristics that we can see in the human species, particularly having to do with our cognitive abilities and our hypersocial abilities, or the evolution of the most complicated network that we know of, and that would be the human brain. But indeed, comparative genomics has been informing us that something happened recently in the evolution of humans associated with a lot of activity of genetic parasites that we considerably just dismissed as transposable elements. But if we can step back and cast this concept from a different perspective, in which we examine what the genetic parasites, what the viruses do first, and then consider the consequence to the host, I think we can develop a different perspective in terms of the relationship that the viruses have with their host. And this allows us then to introduce these ideas that a virus-first perspective allows us to examine a distributed set of events that are inherently capable of promoting the creation of a network. They're inherently functional as a population, and they're inherently cooperative. And so the concepts that I really want to introduce today are basically two. That one is the issue of addiction, is that when you get colonized with a stable genetic parasite, it's not an accident. It has a strategy that ensures its maintenance, and that strategy can actually have consequence to the survival of the population that's been colonized. And two, that these most selfish of all entities, viruses, can't get any more selfish than that, are really the entities that have identified for us the foundations of cooperative molecular behavior within a population. And this has to do with the emerging concept of quasi-species, at least the modern version of it, not the historic version, which was based on Neo-Darwinian thinking. So today I'm going to have too many slides to present, and each of these slides has too much data for me to really get into or try to explain in any detail. But my objective in sort of rapidly going through this kaleidoscope of slides is to just to sort of paint the image of the relevant evidence, the relevant concepts, and the relevant arguments that relate to this thesis. And I'd like to end it with the cover of a book by a colleague of mine. The hypothesis basically is that of virolution. So human attributes that we would try to explain would seem not very amenable to the action of viruses: the emergence of a large social brain, the complicated cognition necessary for language to be able to happen in our brains, and the complicated associative process and extended social bonding. We are a hypersocial species, and the emergence of our phenotype, particularly with our large social brain, is what endows us with this capacity. So really what we're interested to focus on is the origin of social bonding mechanisms and whether viruses had anything to do with that. And we will be examining the origin of the big social bond, the maternal bond, as happened in the emergence of placental species, as a way to understand some of the underlying mechanisms and the process that might have worked for the evolution of human. What we're really confronted with is trying to explain the origin and creation of networks. Historically, Neo-Darwinianism has sort of asserted that if you have a random variation that happens to be useful, it gets selected for, it gets locked into the genome, and then you undergo another random variation that happens to be useful, gets selected for, gets locked into the genome, and by this step-by-step process, somehow we create a network eventually. There's reasons to think that fundamentally that can't happen, and actually philosophy of science has something to say about that, and I will mention that a little bit. And in fact, the assertion that I will be presenting, or the concept, is that that's really not the way it happens. That networks tend to be created by colonization by a population. That population has some coherence to it. We can understand that coherence from the concepts of quasi-species, at least the modern concepts. And this allows us to understand how when we see what looks like massive destructive events, like all the indels that have happened in human evolution recently, that they really aren't massive and destructive, that they're part of an agency, of a population of agents that has superimposed onto its host a new system of identity, and it uses addiction modules to do so. So basically, we're talking about the emergence of regulatory networks, and it's been accepted for some time, even though the evidence for this wasn't good, that in order to explain the emergence of the human phenotype, the large, complicated social brain, we need to understand how we've created a regulatory network that is significantly more complicated than what existed before. So we come down to the issues of regulation, not coding genes, and we're really trying to understand how you can edit in a coherent way, in a complicated way, in a dispersed way, something as complicated as a brain, and end up with something that still works. And the assertion will be that this is an example of virus-mediated evolution. So a simple definition of a virus is just a parasitic genetic entity that depends on its host for replication and or propagation. Amazingly, the definition of virus has undergone significant changes in the 2,000 years it's been in use, and it's still susceptible to some change in terms of what meaning is. But this simple definition has some profound implications already associated with it. And one of them is that a virus has to be competent and capable to operate with host code, but it also has to bring new code that allow that host to make virus or persist or replicate or lyse as a consequence of virus. So it's inherently competent in code, and it's inherently competent in extending code. So this makes it sort of a fundamental entity able to extend code, but able to extend it in a distributed way, in an inherently network-based way. The reason this line of thinking wasn't pursued historically is because there was no mechanism or no reason to understand why a virus would want to do anything like that. We think of them simply as destructive genetic parasites, and we don't have any underlying explanation as to what would drive symbiosis, for example. And if you think about what the problem of symbiosis, this is the merging together of two organisms that have their distinct instruction sets, they have their distinct code. How can we coordinate sort of the instruction, for example, of a cyanobacteria with whatever host happened to be colonized with it? We have a problem that's one of significant code editing and how to cohere these two systems. I think we can see that viruses provide a potential solution to this. But one of the problems is that we always sort of think of a virus and think of how they act in the context of the individual agent. Up on the screen are a whole pile of ideas that are inherently related and, in my opinion, really can't be separated. Because once a virus has colonized its host in a persistent way, and we're all colonized with a whole array of viruses, both some in our germline and some that are epigenetic, then we can call that state symbiosis. But symbiosis, being colonized with a virus, recognizing that virus as the same or different, we get into issues of what is self-identity, what is immunity, what are the principles that can allow something to be selected at a population level. So in my view, all these concepts are basically network concepts that we tend to approach in a serial way, in a Neo-Darwinian way, one phenomenon being discoupled from the other. And one of the ideas I hope to implant is that really they're not separable, because network-based concepts do not reduce to sort of serial ideas of selection and counter-selection. So what we're going to examine is what has happened recently in human evolution. And we already know from the sequencing projects over the last 10 years that the open reading frames are surprisingly similar between us and our recent sort of species cousin, chimpanzees. But it is known that there is a significant difference with respect to the occurrence of these insertion and deletion regions that were called indels, and that the number was around 68,000 the last I looked at it. And I was reading a paper last night that indicated that about 30,000 of these indels are really retroposon, LTR-mediated or endogenous retrovirus-like elements that are active to a considerable degree in the human brain. And we're going to be talking about that. But just step back and think about this observation in and of itself. How can we evolve the most complicated organ we know, the most complicated network, with this type of destructive colonization? If we just think of these things as simply random mutations and insertions that for the most part are junk and are damaging. How is it possible in the background of such damaging phenomenon to create the most complicated organ we know of? And my assertion is that the thinking is what's the problem. But if we look at it as an agent-driven colonization, which is superimposing a different identity system onto its host, we can start to understand why there is a selection for this to occur. And what we're really trying to explain is basically the doubling of the human brain that occurred from its Homo erectus predecessor, which was about the size of a chimp brain as well. And particularly interested in understanding the emergence of those regions like the frontal cortex or the visual cortex or the decrease of the olfactory bulb and the various other changes that were coordinated and associated with human evolution. How can we explain the underlying selection, and why would a virus have anything to do with this kind of phenomenon that seems completely out of character? And we can acknowledge that soon after birth, there is a considerable growth that occurs in humans that we don't see in the other primates, and this is associated with also the acquisition of language and the emergence of a variety of human cognitive skills. During this process, we're seeing massive apoptotic pruning occur of the neurons. Apoptosis is a phenomenon of self-destruction, which in its origin is thought to be associated with an immune system against colonization with various viruses. I won't have time to get into the details of this particular line of argument, but I can just point out that the apoptotic system, at least in the human, was substantially modified by the action of LTRs and viruses during recent evolutionary times. So we're really interested to examine regulatory networks and how we accomplish growth transformation, how we can do massive reprogramming of invasive and learned connections, and why there should be any selection at all for humans to operate in groups to have extended social bonding that has consequence to their existence. And we're going to examine mechanisms of social bonding to see if there are indeed viral footprints there. So we already know that in the human genome, something like 8% of our DNA is LTR-containing material. That means it's directly related to the action of various families of endogenous retroviruses. And in the case of human, the most active one recently is the HERV-K family, which in itself is a complicated set. This is quite a bit in excess of what we have. So we have a lot more endogenous retroviruses in our DNA, by a lot, than we have coding sequences. And much of this is different with respect to our closest relative. We're going to come back to that point later. But this is a perspective we're going to try to pursue. If we start off with a sentence definition of a virus as simply as a genetic parasite dependent on its host, and acknowledging that it has to be competent in host code and has to extend it, it is from this perspective we need to understand why such an entity is capable of creating complexity onto its host. This is the usual life cycle of a virus, at least as we understand it. It's a lytic virus binding to the surface of its cell, uncoding, releasing its genetic material. That material either makes it to the nucleus or stays in the cytoplasm, depending on the strategy of the virus, in which it can integrate, get transcribed by host machinery, translated, and assembled and transported. And in the context of some viruses, for example, retroviruses, that internalization process actually creates, at the same time, its regulatory coherence, because it creates the LTR associated with the expression of the gene at the time of its insertion. Over the years, we've come to realize that there's a lot more virus on this planet than we had ever understood. And this comes from a variety of studies of various habitats, but pretty much every habitat that I'm aware of that's been examined is predominantly viral. This shows a drop of water, ocean water, on the fingertip of a person being magnified under fluorescent scope, which looks at the presence of DNA and or RNA, and then being magnified even greater in an electron microscope. And we've come to the conclusion that viruses are present in enormous quantity and pretty much always have been in all the habitats that we've examined. And actually this number is an underestimate because more recent numbers indicate that there's even a larger number of RNA viruses. This was a number based on DNA viruses. By the way, that number turns out to be 10 to the 31 virions in oceans at a particular time, which happens to be, if you put them end to end, 200 light years. I'm sorry, it's more like 200 million light years, which is a quantity of virus that's mind-boggling to think about. What this informs us is that all life is a survivor of this habitat. If we remove the virus from the equation, such as when we have inbred mice, for example, in Roman laboratories downstairs, we create a circumstance that is unrealistic and sort of misidentifies some of the fundamental selections for existence of life. And we'll come back to this point because it's very realistically demonstrated by anybody who grows mice, and I'll be talking about that later. So the main assertion is the colonization with these entities requires a strategy for the maintenance of this information, and that strategy has consequence. Viruses aren't necessarily warm, fuzzy things. They do compete with themselves and with each other, and they even compete with self. When an E. coli is colonized with lambda, for example, it expresses an immunity module that prevents other lambda from colonizing that same E. coli. But it also has changed the potential of that host in a variety of ways, which I will sort of present as the emergence of what we call group identity. So addiction modules is the concept that I'll be developing a little bit, but even more so when I talk about the origin of the immune system on Thursday, are one of the consequences of these colonizations, and it can be direct because the virus actually encodes genes that are capable of destroying the host if they're not protected against, or it can be indirect, and this is the more generalized version, where just being colonized with the virus means you're immune to that prevalent virus, and if the habitat has a lot of it, which we know it does, then that really changes the selective advantage for a population that's persistently colonized. So the main thesis is that being colonized, either in your germline or epigenetically, it sort of doesn't matter. They both can result in a population that's been marked by the virus as distinct, but that population now has some capacity to outcompete any population that's not similarly colonized. So it's gaining, it's a net gain of systems that regulate its sensitivity to other viruses and therefore sort of promote the origin of immunity in the system that is so colonized. I've recently published a paper that sort of describes this argument in the context of prokaryotes and eukaryotes. But what I really want to do today is emphasize a phenomenon which is down here, and that has to do with the existence of quasi-species and how this relates to the concept of cooperation, and why this identifies really a very fundamental process that relates to how molecules interact in cooperative ways that we had previously not considered. This is my version of the tree of life, and it's colored because it represents the activity, the dynamic activity of viral genomes within their host. And on the left side are the prokaryotes with the archaea up on top and the bacteria below it. And the assertion being made in this image is really the dynamic component of all domains of life is predominantly material that's originating from virus action. It's either direct and intact proviruses in some cases, or it's pieces, bits and pieces of what we call defective viruses, or prophage, or cryptic prophage, or LTRs, or ERVs, or transposons of various types, mostly related to the action of virus. And you'll notice at the center of this is LUCA, the last universal common ancestor, which is depicted here as predominantly a viral-like entity. And this has to do with the concept that LUCA was an RNA entity and fundamentally had to be a quasi-species entity and consequently had to be a cooperating entity. On the right side are the prokaryotes, the eukaryotes. And the topic we're interested in is human evolution, that little teeny section way down at the bottom right, marked by the arrow. And what we'll be trying to understand is what happened recently in our evolution that might have affected our capacity with respect to extended social bonding, particularly. So for many years, it's been sort of strongly felt that viruses are just pirates of genes. They take material from the host, they occasionally move it from one source to another, and therefore they have really no place in the tree of life. They're just thieves and pickpockets. And this was based on evidence that indicated that they were clearly capable of doing so. But as we've done metagenomics and as we've studied the genomes of pretty much any domain of life, we can really see that the new parts of the DNA, be they human or be they bacteria, are generally a consequence of virus action. So in the 13 years since various E. coli genomes have been sequenced, because of adjacency to tRNA integration sites, we can pretty much conclude that 20% of the material, the variable material that we see in bacteria, most bacteria, particularly pathogenic bacteria, is due to some version from the action of virus colonization. So it's apparent that genome variation is really manipulated to a very large degree by the action of these agents, and in some cases, which I will mention in greater detail, for example, the E. coli 0157, which is the most sequenced and the most studied genome of E. coli, we can actually have some details about the nature of the viral activity associated with that phenotype. So the existence of such sort of ongoing and distributed information colonizing the host from external sources means that the original Neo-Darwinian tree of life would need to be modified to incorporate this cloud of information that exists outside of the germline, but is accessing it and modifying it in these unending ways. So how then might a virus persisting agent affect the survival of its host? And for me, the sort of insight into this came from the studies of phage, particularly the phage P1, which was an issue that was studied by Michael Yarmolinsky in the 90s in trying to understand why when you would mate the phage or cross it or eliminate the episomal phage, the host would die. And hybrid dysgenesis basically would happen. And the odd thing about P1 is that it was studied for 20 years before this phenomenon was sorted out because it was very inapparent, sort of difficult to understand. But basically, with the loss of that phage, we had disrupted a strategy that compelled its persistence. And that strategy involved the generation of a toxin that was stable in time and the generation of an antitoxin that was unstable. So if you had destroyed the presence of the viral genome, the cell undergoes immediate apoptosis like self-destruction. But this has consequence because the presence of that addiction module, the toxin-antitoxin, is not specific only to the P1. It's actually active against a number of other colonizers. So it is apparent that once the host is colonized with an agent that brings this strategy to it, this addiction module, it is changed in its capacity to survive, particularly in a viral habitat, and it is changed in its capacity to compete with other populations that aren't so similarly colonized. So this concept of an addiction module was coined in 1993 by Michael Yarmolinsky in order to explain the specifics of this episomal phage persisting in E. coli. But when you think about it generally, you can start to see that it has a consequence to any population. For example, if I had a persistent infection with something like equivalent to measles, and none of you were immunized, and my being up here talking, making an aerosol of measles virus, would put you all at serious risk of survival. as a consequence. And we know these aren't just theoretical sort of considerations. Anybody who grows any species, be they shrimp, plants, bacteria, or humans, really has to deal with the problems of bringing in a related species that has some persistent agent that can threaten the survival of this other species. So this led to this proposal that was published by me a few years ago, that we can actually propose that group survival, because this is transmissible information, can be mediated by the presence of this transmissible agent. And this is a sort of a generalized strategy as to how this would work. For example, we would consider a population that is uncolonized with a particular agent, and it exposed to that agent that may or may not cause a plague sweep of the population. Sometimes the colonization is without this degree of death, but sometimes it is. The result, however, is that you end up with a population that has been modified by the persistence of a genetic parasite. It could either be in its germline, like an endogenous retrovirus or a cryptic prophage in the case of E. coli, or it can be epigenetic. For example, if we're colonized with a BK virus, or humans have lots of herpes viruses in them, for example, which are lethal to some of the other primates. So this doesn't have to be a genetic colonization, although it frequently is. If that population should encounter another population that hasn't been thus characterized or colonized, we see that this will have a distinct disadvantage to the uncharacterized populations. In case you think this isn't relevant to humans, you might imagine what happened when the conquistadors went to the New World had they not been colonized with the viruses they were colonized with. And for those of you that grow animals in vivariums, such as mice, there's a famous haystack model that tries to understand the relationship of different populations colonizing different haystacks. Mouse hepatitis virus is one of the most highly studied persistent viruses of mice, and if I go out and catch a wild mouse, 99% of the time it's going to be persistently infected and healthy with mouse hepatitis virus, as well as maybe 20 other different mouse-specific agents. But if I take it downstairs to our vivarium that's virus-free, can predict pretty reliably what the consequence will be, and that we'll get reproductive collapse and population collapse of the virus-free population. And this is why veterinarians worry so much about bringing wild animals into the vivarium. So these are realistic problems, my point is. This isn't just a theoretical concern. This is something that relates to anybody who has to maintain a population of any particular species. So now I want to sort of briefly talk about the concept of quasi-species and why was led to the conclusion that viruses have to be essential agents in the root and the tree of life. This is a paper that was actually written by a colleague of mine and myself. Gunther Witzeny is actually a philosopher of science. And his interest in this topic stems from the nature of genetic code and the relevance of language and meaning to whether a biological code is actually equivalent to language. And if you recall, the origin of all the terminology we use in molecular biology, like codons and translation and transcription, these were all stolen from language analysis in the 50s. And he wanted to know, is this an accurate sort of application of these concepts? Because that implies lots of other deeper issues with respect to the nature of code and how you generate or edit it. So Gunther Witzeny is actually particularly interested in biocommunication theory, and he had spent a lot of time, as I did in the 70s, wondering about virus evolution, and we're going to get into this a little bit. This has to do with the origin of the quasi-species model that came out around that time, and he assumed that we could understand the genetic code based on the premises of the quasi-species theory that had emerged in the 70s, and he spent maybe 10 years trying to evaluate that, and basically came to a conclusion that it couldn't work because it was too syntactic. It was too dependent on syntax providing meaning and too independent of the fact that context has a big consequence to the meaning of genetic information. In case you don't think this might apply to the information meaning of a virus, most of us are colonized, for example, with one or another form of herpes virus. That means we have the full syntax of that 100 or 150 kbs of information resident in our cells in some place. That same information in a different context will kill you if that virus happens to get into your central nervous system and you're immune suppressed, or there's damage or not damage, or it can be completely inconsequential if it happens to be in a neuron and the latency associated transcript is turned on and keeping that thing in a latent state. So the syntax of that big piece of virus information is completely dependent on context. And this is not only true for viruses, this is generally true for genetic information during development, for example. It's clear that how information gets accessed and how its meaning gets implemented in terms of phenotype and code matters a lot. So this is not an issue that we can just dismiss. It actually matters for how multicellular systems work and how viruses work. But the quasi-species theory that we had both been pursuing for some time, which was originally implemented by Manfred Eigen in the 70s, basically is a Neo-Darwinian view of evolution. It assumes that the master fittest type is the most fit version of, say, an RNA genome, and he used an RNA virus as a way to develop the quasi-species equations, and assumed that there was a high error rate, and this would generate a population of molecules quasi-species, and that we were dealing with this issue of maintenance of information content and whether it would decay when you had too much variation. So this is the ordinary differential equation that was the expression of a quasi-species based on the survival of the individual of fittest type and based on the generation of error when that thing copies as an RNA. What followed, however, was a whole series of experimental evaluations beginning in the mid to early 90s, initially in the laboratory of John Holland, but also Esteban Domingo and many others. And it culminated, in my opinion, with this paper that was published in 2005 by Raul Adino's lab at UCSF, but there were similar papers that were published by Carla Kierkegaard at Stanford and subsequent papers by Esteban Domingo's group in Madrid. And they came to what in my judgment was a startling conclusion that quasi-species really don't operate by the survival of the fittest individual type, that they are fundamentally cooperative in terms of how they attain fitness. And the experiments that were done in this paper were ingenious in my view, because they actually were able, because it was done with poliovirus, we had a level of control of the virus that was unattainable in most other systems. It's possible to make a version of the polymerase, for example, that has high fidelity and low fidelity. It's possible to clone out the consensus sequence. It's possible to reconstruct it. It's possible to test a lot of concepts with respect to whether there is a master fittest type or whether diversity per se has to exist in order for this to attain fitness. But the experiments, I think, were quite clear, although mainly ignored. So the summation of this line of thought was really put together in a very comprehensive review that just came out this month by Esteban Domingo's lab. And this, for those of you who want to read it, it's about 900 references, but it is really a tour de force with respect to the history and the experimentation underlying quasi-species theory, at least current quasi-species theory. But the main point is sort of made that when you pass these populations as populations, you gain fitness by passing it as a population, even if the consensus sequence doesn't change. If you clone it out into an individual type of molecule, fitness is always lost. Now, historically, evolutionary biologists dismissed this entire line of thought as basically misunderstanding of fish or population genetics. But in fact, the experimental evidence really makes a strong case against that. And this is just one example of the kind of experimental evidence that exists, but there's a lot more to it than that. And basically, if you take a quasi-species, sort of this complicated population, and you measure its fitness by a variety of parameters, and in viruses you can use very quantitative measurements of, say, replication or something like that. And you clone out all the individual types that are present, they're always of less fitness in the mixture. Well, this tells you something that some kind of cooperation or interaction is happening, but it's even a lot worse than that, in that some members of this population are actually what we call defective interfering. And that's shown on this map of sequence space, where you can see that we're actually going below the plane of selection. And it's possible, and it's been shown in some cases, that you have a member of the population that actually interferes with the replication of the wild type. But that interference is important for the phenotype of the population, for example, persistence or a vaccine strain or a variety of circumstances in which you have combined interaction of what you would consider historically a very fit and a very unfit, not only unfit, but actually negatively fit member of the population, so that you end up with something that is functioning as a population, and you can break it down into these various subsets. And the thinking is what happens is because of the information in the content of the virus is not suitable for solving all the problems, that these populations are able to segregate phenotypically, at least in various instances of virus replication, and you end up with something that's working cooperatively in solving populations. This is an experimental result, not a theoretical result. But it leads us to a concept in which we can start to propose at this very basic level with respect to operation of RNA molecules, how cooperation works. Now we have to remember that LUCA was thought to exist, the last universal common ancestor, in an RNA world. And that would indicate that it was fundamentally cooperative if we apply these concepts from these kinds of quasi-species measurement. The rather amazing point about this is that as you pass a quasi-species, for example, an RNA population, the fitness is always changing whether or not the individual fittest or the consensus type changes. So if you take this population here, for example, and compete it with this population there, this one always wins. And this isn't simply a theoretical concern. For example, a person who gets a liver transplant, who gets a liver from a patient that had hepatitis C virus, we see these kinds of phenomena occurring in the clinic. Within a matter of a couple of weeks of the transplant, a prevailing quasi-species will displace the population that existed before. So these are sort of observable things. So here's the conclusion from Esteban Domingo, that one of the sort of main features that has been measured over all these sort of types of experiments is that these genomes, these numerous variants, are bound together, even though they have extreme rates of mutation, into these co-selected partnerships. And so we end up with a population in which the population is where the fitness can be seen. That even if you clone out the fittest type, it cannot compete with the population. So this identifies a fundamental process where we have mixtures working in a network-like circumstance to attain some type of identity of the population, and that diversity itself is essential for this to happen. So we now start thinking in terms of consortia and networks, not just individual types. So this is not an idea that's popular with evolutionary biologists, but it is one that I think is pretty well supported by the relevant evidence that I'm aware of. What this tells us, however, is when we examine what has happened in the colonization of a germline, for example, our human DNA looks like it's been colonized by a quasi-species of agents, not just an individual agent. We have to examine things from a network perspective to try to understand what is the consequence. So here's the concept then, is that a genetic parasite population can stably colonize its host and alter its identity as a consequence, and alter its survival with respect to competing populations, and that this is the acquisition of complexity and the acquisition of identity, and it's network-based. It fundamentally has to superimpose these mixtures in a diffuse way. Now we start to understand some of the big ideas that have come forward in the last 10 years with respect to what viruses might have had to do with human evolution. One of them has been, particularly from the French group, and the discovery of the giant mimivirus that encodes 1,200 about genes, is that we might be able to explain an alternative origin for something as complicated as the nucleus. And what you're seeing here is a structure in an infected cell that was what we call a replication center, a replication factory for the mimivirus. And the first time it was observed by cytology, it was mistaken for a nucleus in that cell. Beyond that, though, Patrick Forterre, particularly at Pasteur Institute, has sort of submitted a variety of other provocative ideas, and possibly the most provocative is that we can explain the origin of DNA itself and DNA replication apparatus itself if we invoke the operation of viruses at various stages. I'm not going to discuss this further or really elaborate on it much, just to point out that there exists some very big ideas with respect to what viruses might be doing with regard to host evolution, and they're very fundamental with respect to their implications. The issue that I want to examine in a little more detail is whether or not recent human evolution, particularly in the context of phenomena that affect extended social bonding, might have been modified or mediated by the actions of these genetic parasites we call viruses. So we do know that there exists in the human genome a population of agents that are related to each other by the action of reverse transcriptase, and they're shown here as lines, signs, and retroposons of various types, either having complete or incomplete genes encoding for the various endogenous retroviruses. In some cases, only LTRs are apparent. The lines and signs are themselves the products of reverse transcriptase, and there's frequently good evidence that there's interaction occurring between all these agents, although I'm not going to discuss the specific evidence relevant to that. But what I would point out is when the chimpanzee genome was first sequenced in 2005, and sort of buried in one of the tables, was this interesting difference between the human and chimpanzee DNA with respect to the nature of LTR-based agents that we find in them. They're basically distinct classes of retroviruses in chimpanzee versus human. It's the alpha, the beta, and the gamma class of retroviruses. And so it hadn't been addressed at that time, or was really kind of ignored until recently, whether this might have some regulatory implications with respect to the emergence of brain function and the changes in the social dynamics. So if we look at the evolution of mammals and placentals particularly from the perspective of virus action, one of the things that really interests us in terms of mechanisms of social bonding and mechanisms of social interaction happened with the invention of the placenta and a mother that had to bond socially to its offspring for extended care. And it was interesting that when you examine reproductive tissue, particularly placental tissue and other associated tissue, that we see this preponderance of high-level expression of a whole range of species or lineage-specific endogenous retroviruses that are highly expressed in the placental tissue at that time. This is a picture that was taken of a human placenta in the 70s by the J. Levy group. And what it's showing is the production, the high-level production of these particles, these endogenous retroviral particles. They're not actually infectious, they're defective. And most all of these viruses from most all of these mammals are defective, with the possible exception of sheep. But they're being expressed in vast quantity. You see the numbers on the scale of 10 to the 6 per cell. And so the question being, what's going on? Why would the emergence of the placenta and the emergence of sort of the mammalian lifestyle be associated with so much activity with respect to the placenta? In the 90s, in the late 90s, I was contemplating this issue and suggested that viruses really provide a solution to one of the fundamental problems that live birth creates, and that is the embryo is antigenically distinct from the mother. And in order for the mother not to reject the embryo, its immune system has to be modified in a very fundamental way that will allow the embryo to be supported even though it's antigenically distinct. and that we knew viruses are particularly good at modifying immune function, and they would just provide a convenient way to modify the host to allow the emergence of placental biology and the maternal sort of relationship. Since then, there's been a whole array of studies that have found that indeed some of the fundamental functions of the placenta are mediated by the action of endogenous retrovirus, such as its incisions 1 and 2, associated both with the fusion of the trophoblast and the immune suppression that happens in the host. But I think the most compelling experiments along these lines were done by the Pomerini and Spencer group in Texas and elsewhere, in which they were evaluating the consequence of in that system, it's possible to actually terminate the expression of these endogenous retroviruses and look at the consequence to the biology of the host, and it was apparent that it's essential for implantation. So in this particular instance, we can clearly assert that the virus colonization in this sort of complicated and diffuse way was essential for the biology of the host to emerge. But what has come to light recently, beyond that, is that the related transposons apparently were involved in a rather extended rewiring of a regulatory network of the placenta. And this is a paper that came out recently from the Wagner group in Nature Genetics. And the real point that's being made here is that we see 1,500 genes were modified as a consequence of colonization with these LTR-expressing endogenous retroviruses that created sort of a new regulatory paradigm associated with the emergence of the placenta. Now, the reason this is of a special interest is because the placenta is also associated with the activity of the social bonding through the mother, and that this had to be mediated by part of this network change at the time of the emergence of the placenta as well. So this leads us to the question is, how do mammals identify and bond to their offspring, and was there any evidence that viruses might have had anything to do with this? Those of you that are in this topic, of course, will know the work of Larry Young in Atlanta that has been particularly active in exploring the mechanisms associated with rodent bonding and pair bonding in the prairie voles. I'll mention that in a little bit, but I just wanted to summarize sort of a feature that we've known for some time, that there exist in all tetrapods, except for the higher primates, this organ, vomeronasal organ, that's found in the snout, which is distinct from the receptors for the main olfactory epithelium, shown here, and that they're associated with establishing social bonds, both with mates and with offspring, and in terms of sexual reproduction. This has been evaluated in a variety of ways. It's possible to cut the receptor neurons, which go to different parts of the brain than the other olfactory receptors. And there's a connection here actually to the function of the placenta, because the Bruce effect induces an inflammatory reaction in the embryo, which causes it to be rejected. Now, these are phenomenon that did not carry over to the old world primates. And this is associated with a genetic colonization that I'm going to talk about in a little bit. But the model that Larry Young used to sort of sort out the mechanisms of this social bonding were these prairie voles, where you have both the montane vole and the prairie vole. One is bonding, the other is promiscuous. And I'm not going to sort of get into the details of this experiment, but basically what we're showing here is difference of expression of the vasopressin receptor associated with addiction centers of the brains of these animals. And if we examine the underlying genetic change associated with the speciation event, we see that the regulatory region has been colonized with a genetic parasite that he called a microsatellite. Microsatellites are usually thought of as resulting from strand slippage of the polymerase, but the changes in these two regions I don't think can be explained by strand slippage, because they're too complicated and they're not associated with one particular domain. They're scattered in a larger region. Alternative, which I think makes more sense, is that they're the products of unequal insertion and exertion of endogenous retroviruses, which was one of the original models of Young and explaining the origin of the microsatellite. So what we see here is that behavior, regulatory changes that are associating social bonding of behavior are being manipulated by the action of parasitic sequences, and this entire bonding can actually be achieved with a virus that is built to express the same receptor. So can we translate any of this to the evolution of a human phenotype in terms of thinking about the neurobiology of pair bonding or extended social bonding that happens in humans? Before sort of touching on this topic, it's worth noting that with the evolution of the old world primates, we did have a significant colonization event in that all the 7 genes associated with the vomeronasal organ receptor got turned into pseudogenes as a consequence of genetic colonization. And at that point is when we had a major invasion of endogenous retrovirus in the primate lineage. Could these two facts be related? Here are the colonization events that we can see occurred at the various points with respect to the primates and more recently with respect to humans. Interestingly, if we examine in a greater detail, we see an association, but certainly nothing that we can call causal, with respect to the colonization of the germline. So for reasons that we would like to explain, a mechanism of social bonding, the vomeronasal organ, which exists in fish, by the way, and has been preserved in all tetrapods and is crucial for the survival of all these animals, was taken out on the great apes. And as a consequence, a different mechanism of social bonding, probably not the underlying process of the receptors and the addiction centers, but the input must be different. It's no longer olfaction-based, it's no longer being part of the major histocompatibility locus, which was part of those receptors. If we look at just the crude differences between human and chimpanzee chromosomes, such as shown here in the cytogenetic report, we can see regions that are clearly distinct. And these are called C-bands that are stainable. And what's present in these C-bands is actually numerous copies and defectives and repeats of the endogenous retrovirus HERV-W and HERV-K. So we do see that humans and chimpanzees have differed considerably as a consequence of this colonization event, but this has, for the most part, been dismissed as having no consequence since it's junk DNA, particularly since much of it's on the Y chromosome. Yet if we look at patterns of expression of the transcripts for the information, again we see not only some of reproductive tissue, but we see a specificity for brain expression, particularly in the neocortex. And here's a summary of another study that evaluated these expression profiles. And the last sentence emphasized in red. Most striking is that while the majority of verbs analyzed in this study are expressed in the human brain, almost none are expressed in the old world monkey. Brains are the only weakly expressed in the context of the old monkeys. Why would that be? Why would we have our brains colonizing and expressing this destructive entity that somehow led to the emergence of the more complicated cognitive functions that we have within us? So the assertion will be that, in fact, group selection, group identity does exist, and it's been promoted by the action of these viruses, and that in contrast to pretty much all the existing mammals, particularly the tetrapods, humans lost the olfaction-based link to group identity and to bonding, and therefore had to develop a really distinct process. And I would assert that that was probably cognitive in nature, that was associated with learning, that was associated with language, and that's associated with how we form our bonds. So they're much more cognitive, they're much more based on visual and audio input, language-based input, and that these then define a mind that emerged susceptible to forming addiction modules but that they're based on these cognitive. So in this case, Young and Wang argued that even in the context of human, you see the same addiction centers being activated with respect to human social bonds. And we see the same kind of mechanisms that were visible in the voles. So we might then consider whether the actions of these viruses in taking out these historic systems and transforming the capacity of the brain to form a more complicated and regulatory-based network underlied the evolution of the hypersocial brain that we now have. And although we can't obviously conclude that that's the case, certainly we see the footprints with respect to virus activity. So these are the conclusions I would lead you with respect to my thinking in terms of the evolution of the higher functions of human social bonding. And I would just like to leave you with an image of the cover of a book published by Frank Ryan last year in terms of how we might start thinking about what viruses had to do with the evolution of life and this new concept that we would call virolution. Thank you.
So, good evening. I would like to begin by thanking the organization of this meeting. It's a real pleasure to be here and to moderate this discussion. And I believe that for most of the people that are in this room, you have changed the way you see virus and the notion that viruses are only the carriers of disease and death. It's absurd, at least, because the viruses are essential part of evolution of the species in this planet and gave rise in some way to humans. I think that there is a lot of questions in the audience, so please don't be shy and put these questions. Professor Villarreal will be, please. Please wait for the microphone.
But I think we cannot find endogenous retrovirus in plants, or why is that, do you know?
If you recall the image I projected about the tree of life, the kind of orange one with the blue tips, I didn't discuss it, but labeled on each of those domains was a peculiar feature that wasn't emphasized, and that is all domains are peculiar to the kinds of viruses that colonize them and create their identity systems. For example, bacteria are particularly prone to prophage, to large DNA viruses, double-stranded DNA viruses that come in and bring gene sets as cryptic phage and mixtures. That doesn't exist hardly at all in eukaryotes. There are some examples of large DNA viruses integrating occasionally in eukaryotes, but for the most part, that doesn't happen. The relationship is quite distinct. We have a lot more activity of retroposons and endogenous retroviruses. So every domain you look at has its own sort of peculiarity. Rodents, for example, make endogenous retroviruses as viruses. Primates don't seem to. Plants are very susceptible to the action of plus-stranded RNA viruses, something that you don't see at all in archaea. So you can't generalize too much about the nature of the virus to the host because they're all sort of connected in a very peculiar way.
Please.
Waiting for the microphone.
Well, a number of questions. First, I'd like to know whether all viruses that we import, that humans import from outside. They're penetrating in humans' body, are cooperative in the sense that you use this term and contribute in this way to evolution, to human evolution. Second, when the cooperative viruses, it seems to me, I understood from what you presented, that they keep their competitive behavior too. That they must keep in order to maintain their own colonies in the organisms they penetrate. Is there some way to measure how much cooperative they are and how much competitive they are, and how these two parameters are related to each other? Well, I'll satisfy myself with these two things.
That's actually a very excellent point, I think, because it sort of gets at one of the underlying concepts that's context dependent. For example, I mentioned that wild mice are frequently colonized in a persistent way with mouse hepatitis virus, which is one of the highly studied models, and it's true of a lot of other ones. That same virus and that same host can have a very different relationship depending on the context. If you've colonized an island with a mouse that happened to have been free of mouse hepatitis virus, and you reintroduce another mouse that has it, you will wipe out that colony, and that's been observed experimentally, in the field. So the relationship of whether it's cooperative or destructive is conditional. It depends on whether it's attained persistence in that population. So persistence is really the relationship that identifies cooperativity. But persistence relates to a whole array of circumstances, how the virus is being introduced, if it's being passed from the mother to the offspring, if the mother is immune. There's too many conditional variants to give an absolute answer to that. It's more like asking for the meaning of a word in a sentence. That meaning is going to really depend on the context that you put it in. So this is a process. You get back to a circumstance that basically identifies context dependence as a fundamental aspect of genetic information. Remember, the quasi-species, as you pass it, it changes its fitness with respect to competing with prior populations. So there's no way to predict the absolute fitness of that population until you put it in competition with some existing or pre-existing population. And you can imagine, for example, in the context of human and recent human evolution, what if we had exterminated all chimpanzees last century? Then that means there was no possibility for HIV to have emerged as a human-specific disease. So that context is completely distinct. There's no absolute prediction about it. Well, the syntax also can be context dependent, because we just flip it over, then it becomes antisense, and it actually inhibits what it was coding for. And that happens. And there's examples of that happening too. So yeah, this gets into the deep issues of the nature of language and meaning, which I wasn't trying to get into at this point.
I have another question. Sorry. About the haystack model that you talk about here,
Where's
okay.
this?
Sorry.
Oh, here. Oh, wait. I see them action. Okay.
Okay, I'm going to be fast. In fact, I read your paper like 1 year ago or something, the paper in Current Opinion in Microbiology, the 2009 or something. You talk about that paper now. And at some point, I think you wrote something like that we don't need kin selection. I'm going to read to understand this group selection. And you were saying more or less the same thing here. I want to tell you that, in fact, I don't agree with that, because, in fact, you are talking about kin selection when you give your example, when you explain why. Because when you talk about the virus and the murine virus in the mouse, in fact, they are, from the point of view of the virus, they are kin of each other. So this is kin selection again.
Well, I disagree with that. And the example that I just gave, for example, with the mouse hepatitis virus, in that you can have completely different outcomes, same virus, same host, in which it's either supportive or negatively selective, depending on the relationship, not depending on the genetics of the virus and the genetics of the host. I don't see how you apply kin selection to that situation. But these can be identical. They can even be offspring of the same parents, and you can still establish this difference depending on the relationship. For example, think about the P1 relationship with E. coli. It can either be lytic or it can be persistent. That outcome depends on the relationship of the virus and the host, not on the genetics of the virus and the host. So in what way would you apply kin selection to that situation?
So I apply kin selection looking to the gene which I'm interested in, which is the virus itself. So it's, we can discuss this by email.
It basically comes to the concept of a lysogenic population, right? If we have two populations of E. coli that are identical,
exactly,
and they even are dealing with the identical virus, but it's whether or not that virus is in a sort of immune state versus an lytic state, you have very different outcomes with the same genetic composition of both.
But in the lytic stage, they are going to be dead.
But I haven't complicated it yet because it gets a lot worse than that. Typically, the situation isn't just one-on-one. Typically, if you look at, for example, the composition of an E. coli that has cryptic phage, you're often talking about the interaction of something like 6 or 7 cryptic prophage that gives you one particular phenotype. And that dynamic is really flexible depending on what happens to be in the sewage water at the time. So I don't see how the concept of kin selection applies to that circumstance.
Okay, I write an email to you. I don't want to
I've had this
monopolize.
conversation. I'd like to continue it, yes.
Okay, thank you.
Please, is there another question?
Okay. In the beginning of your communication, the presentation, you have referred one thing that's, for me, is really important. That is the problem of symbiosis and how two species can coordinate the different genomes. Can you develop a little more about this idea?
Yeah, thank you for that question. So the question is, how can a virus promote a symbiosis between different lineages of life? And we actually know that there exist many examples in which viruses have alternate hosts. And therefore, they have to have the regulatory competence of two different lineages of hosts. So what you would envision then was that a virus that's competent in, say, 2 different kinds of cells can exert regulatory compliance on both those cells and compel them, if it's persisting, compel them into a relationship because of that regulatory compliance. So it's the only entity that has the capacity to superimpose that kind of regulatory paradigm on 2 lineages that were otherwise separate. And for me, if you look at, for example, you look at a chloroplast or you look at a mitochondria, for reasons that I find very baffling, the polymerase associated with both of those looks like the polymerase from T7, not the polymerase from any bacteria. And to me, that's a footprint that a virus was involved in that symbiotic event early on. but it's usually an ignored fact.
May I put a question too? So you believe that mimevirus could be the origin of the nucleus of the eukaryotic cells?
Yeah. The question is whether I think mimevirus could be the origin of the nucleus of a eukaryotic cell. I don't actually suspect that exact virus, and in fact, it's apparent to me that it wasn't a single virus, that it was a consortia. My suspicion is that we had a colony, we had a community of cells that probably involved both alpha proteobacteria and maybe a cyanobacteria that was exocolonized. Something actually colonized the whole thing, and it used retroposons to gain control, regulatory control, just like we see in our brains. So with the origin of a eukaryote, you have the acquisition of a completely distinct replication strategy, which is probably viral, but you also have the acquisition of massive invasion in both the plastids and in the germline of retroposons. And so I think you had this consortia of agents enslaving a community and surrounding it with grease and taking the ribosomes out. And, so it was a much more complicated symbiotic event than the way most people are thinking about it.
Very nice conference. I really enjoyed it. I wanted to ask you something, because you put norm or routine concepts that in other contexts are really provocative. And one day I'm studying the interrelations between the bacilli of tuberculosis with the host cells. And I was talking with a medical doctor that usually take care of people with suffering from TB. And I was asking, and I was telling, since 1/3 of the world population is infected, that must be a symbiosis instead of a sickness. What do you think? And he told me, come on, you should be more really provocative. So like viruses, also bacteria, do you think that the whole bacteria can help in the evolution of species?
Well, there's an emerging concept with respect to the function of the immune system and the bacteria of the intestines and that relationship, as all that is sort of tied together in a much more intimate way than we had previously envisioned. But that's still a relatively vague set of ideas. With respect to the mycobacteria, is fascinating because of the close association that's with the particular host. And whether or not you have phenomenon associated with bringing different hosts together, and you have the jumping of a species and the creation of a disease version versus the existence of a non-disease version. These are ideas that I find fascinating, but I just don't know enough evidence to answer that one way or the other. It does strike me as a possibility that anytime you're persistently colonized with an agent in a stable way, you have the capacity to harm populations that don't have that same stability. So in that context, a mycobacteria persisting normally in a particular host population would provide a huge selective advantage to a version of that population that gets disease from the same agent. And in a sense, this concept applies to any form of communication that you might consider stable. Any transmission that happens from one network to another network that can persist and harm competing networks gives you the basis of selection for network membership. So when we think about the security of a computer network that's distributed all over the planet, and the fact that we're always struggling with reproduction of files that we call viruses, which is logically equivalent to a biological virus, there's a reason that networks are never free of this issue, because I don't think it's possible to specify information content and information reproduction in a way that precludes all reproduction that's going to be disallowed. And therefore, this creates a circumstance for ever-increasing information in the system.
Now, as a scientist, all my works are reported to the reference strain, which is MTB H37RV. But this strain is completely distant from strains that are just come from the body of infected people. What do you consider to be a control strain or a reference strain in any case of study, report, parasite with a cell, a mixed?
That's actually an issue I've been thinking about for a very long time. In other words, if we think about something like influenza, and we're always worried about the emergence of the latest version of influenza, but you actually think about its origins and what should be the reference for our sort of understanding about this virus, you end up identifying the virus that persists inapparently in waterfowl. It's genetically stable, it's transmitted without disease, it's stable on an evolutionary timescale. That relationship is the stable one. The relationship with humans is unstable and changing. It's a dynamic that's always adapting and always changing. So from my perspective, the persistent state is the origin. It's a stable state from which all this other stuff sort of emerges. And if you were trying to sort of label a mycobacterium as a reference, I would find whatever the source is for the persistence of it in its natural population.
I have one question. Any question? Sorry.
So if viruses are so important to evolution, I'm assuming that they have a role in speciation. So what is that role? And if you also said that some type of virus are associated with, are more associated with some type of groups of animals. So if that's constant, can't you do some kind of phylogeny and that virus phylogeny mirrors the evolution of the species? Can you understand my doubt?
That's it. Thank you for that question. One of the problems in our thinking about the virus-host relationship is that we don't segregate the persistent relationship with the lytic acute relationship. And it's really the persistent one that's highly specific. And if we were to, for example, collect persistent virus from this population, we could identify the racial and geographical makeup of this population based on the viruses that you all carry. And that's true of any species that I know of, whether it's a bacteria, whether it's a mouse, whether it's a plant. If you characterize the persisting agents both in the germline and epigenetically, you really characterize where that population comes from. So if you want to type a bacteria, you phage type it. Or if you want to figure out what a mouse strain is, if it's a BELB or JC or a F1 or whatever, you can characterize the Maloney or the other retroviruses present in its germline. If you wanted to say if something is a human or chimpanzee, the ERVs are completely distinct. So we know that this association stands up very well. I don't know of any exception to it, that a species has its own composition. Why that relationship would exist and how it contributes, how this relationship contributes to speciation in itself, I would look at what's happening with the koalas in Australia right now. They're undergoing an endogenization with a retrovirus that looks like it originated from rodents in Africa. I'm sorry, India. You're going to end up, and it's already happening, that the survivors have incorporated this virus as part of a persistent infection that doesn't lead to the lethal leukemia that's happening in many of the members right now. That's already starting to happen. We're already starting to see in Australia the replacement of all the koalas that used to be around with versions that are persistently colonized but surviving the infection with this. However, there's an island, a kangaroo island, that is isolated and has a version of a koala that hasn't been exposed to this virus. The prediction is pretty straightforward. If those populations ever come back together, we're going to see an event that resembles the initiation of speciation.
So only the persistently infected survive?
Yes.
As virologist, I have a question that, well, endogenous retrovirus are important for a lot of things, but how can exogenous retrovirus, for example HIV and HLV, that infect somatic cells, go to a germ cell? By changing the viral protein that interacts with receptor?
Well, you probably know quite well that if you look at the human MHC composition and compare it to the chimpanzee, it really differs notably with respect to ERV colonization, and there's big chunks that are distinct. We're missing some of the ERVs they have, and these are thought to be relevant to the progression of HIV disease. The point being is that we tend to think linearly as one agent, one consequence, one fitness. But in the context of HIV, particularly, we see a more complicated situation. We see the emergence of all these other agents that were completely inapparent to us. I mean, we discovered certain human herpes viruses that we didn't even know existed, but had been with us since before the species formed as a consequence of HIV. So the point I'm making is that you're dealing with these networks of parasites and genetic colonizers that work as a network, and if you disrupt the network, you have consequences. And the consequences may not be because HIV has to integrate into our germline to make a difference. The consequence may be that we end up with tuberculosis, or we end up with a rampant version of some herpes viruses, or some other relationship as part of this network. So we have to start thinking in terms of cooperative networks, not in terms of serial selection.
But you believe that the actual endogenous retrovirus were ancient exogenous retrovirus?
Oh, yeah, I think the evidence for that is pretty strong, that the human HERV-Ks, for example, they look like MLV. They look like they came from rodents originally, because you can't find them in the predecessor genomes of the primates. So yes, I think something happened to us that's a lot like what's happening to the koalas.
Any further questions? No? No, yeah.
I want to go back to the competition cooperation issue. Consider, for example, a football match. In order for it to function, it must have certain doses of cooperation in each of the teams, and also the competition in order to win the match. Okay, so that it seems to me that this can be extended to the genetic material you were talking about, because each of the group identified set of viruses, or the species that they are going to try to attack or to conquer, to colonize, any of these terms, will have both a defensive element in order to protect themselves, to keep their identity alive, and a cooperative element in order to do this together against any invader or colonizer. Now, in the case of football, for example, the teams that are going to win the championship or whatever are those that are going to get the best combination of these factors. In a sense, you can say that there is a sort of evolution there. These groups, if they are going to be prevailing or persistent, as you call, insofar as they succeed to get the best possible combination of these two things. Now, what is the equivalent to this in the system that you are proposing, when you speak only not of groups that have some aim, for example, or some theological tendency to do this or that, and which are, I suppose, the case of the viruses that you are talking about.
Thanks for that question. That sort of leads me to discuss something I wanted to discuss further anyway. First of all, I think we need to apply the concept of agent to these entities. They've often been called elements because they're thought of as random sequences. An agent actually has a purpose and an objective. For example, an LTR retrovirus is really designed as a regulatory agent that goes in and inserts itself and alters existing regulation. So for the most part, these virus populations have this agency character to them. With respect to this feature, the both competition and cooperation, this is the fundamental aspect of an addiction module. In order for like P1 to persist in its host, it has to have both a destructive and a supportive component. It has something that's protective, sort of like a restriction enzyme that will kill invading host, but that restriction enzyme will kill itself by cutting its own DNA if it's not also protected with a modification enzyme. So this is an inherent feature of stable colonization. It has to be addicted, and it has to have both a protective and a harmful component. That harmful component can be turned to the outside and is used against populations that aren't so identified. So that's where you get like the football analogy. The harmful would be they don't have the addiction module, so you destroy them. You compete. But the addiction module sort of compels internal cooperation. That's how I would at least visualize that relationship. And it can be complicated. It's not just two agents can actually work as a component of one addiction module. They don't have to be the same.
Anyone else?
In terms of theory of evolution, do you really think that this virolution is a kind of an alternative to symbiogenesis and to Darwinism? First question. Second question, I didn't understand well what you said about the capacity of the viruses to establish the relationship between two organisms in the case of a symbiotic relationship. Because if the two organisms cannot do this relationship, the virus can. Mm-hmm. And I don't understand. I would like to know what the virus has more than the organism, or less, to be able to do that connection.
Okay. Imagine a unicellular algae like chlorella species, which when it's not symbiotic in a hydra or some other species, is sort of out by itself and available to the mean viral habitat. And in this case, we know there exist large quantities of viruses, chlorella viruses, that are strictly lytic with that species. Once it gets symbiotic in its host, it is protected from the action of the viruses that are prevalent in most water habitats. So this is a sort of circumstance that drives the host to have a sort of symbiotic relationship. But it can be a lot more sort of insidious and complicated than that. If you have a virus that persists in both of these entities, then you can have an addiction module that drives these two together, expressed by the virus, because it will destroy either that lose the persistence. It gets back to this concept of an addiction module as a way to enforce cooperation. So you can think of a chloroplast as a particularly nasty organelle, right? Because it's pumping out so much oxygen, which for species that existed prior to the emergence of mitochondria would have been a very bad thing. So then you start to envision how you can addict an organism by bringing in the toxin and the antitoxin, the mitochondria and the chloroplast, for example, to deal with the oxygen in what would otherwise be a very destructive relationship. This isn't really in conflict with Darwinian evolution or any of the processes. It just expands it on a huge scale because it allows solutions to be generated in the virosphere that involve more complicated gene sets and regulatory paradigms. For example, if you're colonized with 100 genes of a prophage, and we know this happens, for example, in cyanobacteria, if you look at the different populations of cyanobacteria in the ocean, they differ predominantly by prophage colonization patterns in which you have these entire gene sets that have come in with a cassette of sort of pre-selected coordinated genes that are responsive to light in some way. As a component of that, they're also going to be resistive to the phage that are prevalent in that habitat at that time. So what this basically does is it takes variation out of the ancestor and puts it into this huge space and empowers evolution by exponential probabilities.
We could stay here for a long time and continue this discussion. I suppose there is no more further questions, so I would like to thank once again the organization for this excellent meeting, Professor Villarreal for the presentation and for this lovely discussion, and all of you that participate in this meeting. Thank you very much.